Okay, in this video, we're going to go through Competency 12, Genetics, Development, and Human Reproductive Physiology, OpenStax 27 and 28, and Patent chapters 47 and 48. I have links to the Padlet for Competency 12. Again, you'll find several videos that will relate to this competency, and then, of course, a link to the review questions. You'll find both of these in your Blackboard course, as well as contained within this particular slideshow. So we're going to go through and go. All right, so birds and the bees first. We'll talk a little bit about reproduction, and then we'll get into genetics and heredity a little bit. The prenatal period, the postnatal period, and human developmental biology. Of course, the prenatal period begins at conception and ends at birth. And then the natal period, or perinatal period, is when the baby is born. And then, of course, postnatal begins from birth and goes until death. So anyhow, developmental biology generally studies human reproduction and development. We're going to see spermatozoa from the male and the result of spermatogenesis. We're going to see the ova, or an ovum, one typically produced by the female via or through a process called oogenesis. We're going to look at meiosis a little bit. It's a second step to cell division, or mitosis. We know from mitosis, we are going to see our cells throughout the body producing identical daughter cells, exact replicas, 46 chromosomes in the parent, 46 chromosomes in the daughter. That's mitosis. 2 daughter cells typically per parent cell. Now with meiosis, we're going to take a sperm cell, which has 46 chromosomes prior to meiosis, and we're going to break it down into what we call a haploid, or half cell, of 23 chromosomes. We're going to see the same thing occur with an oocyte, or an egg, that is going to go through the process from 46 whittled down to 23 when fertilization has taken place. And we're going to see the 23 from the sperm cell, 23 from the egg cell, combining to form 46 chromosomes. So we're going to look at that as well a little bit. This particular competency, we see there's a little bit of a difference between spermatogenesis and oogenesis. Very, I guess, subtle difference. We will see both going through what we call meiosis 1. We're gonna see the diploid or diploid cell becoming haploid. So we're going to split these in half, and we're going to have 23 and 23. Now, we'll see a little bit later in the next chapter or the next section of this competency later on in this discussion. We're going to see a couple of items or a couple of processes, I should say, that occur that will guarantee diversity among the particular sperm cells and what genetic information they're carrying. So we'll look at crossing over, not that kind of crossing over. Well, I don't know. But anyway, we'll look at crossing over with in regards to meiosis. So anyway, we'll look at that a little bit later. Meiosis 1 happens, and then meiosis 2 is going to divide these even further. So one spermatocyte actually becomes four spermatids, and each of these spermatids is going to be contained, is going to be a sperm cell. So this original spermatocyte became four sperm cells. Each of these are unique in what genetic information they do possess, and it's possible that one of these four could become the sperm cell that fertilizes the egg. So anyhow, so we'll look at this in a little more detail throughout this discussion. Oogenesis is similar. We've got a diploid oocyte, and we're going to get meiosis one. We're going to get what's called a secondary oocyte. So generally a similar situation, just a little bit different terminology. We have a first polar body. The polar body will not become fertilized. So we do see it does split during meiosis two, but these polar bodies will disintegrate in essence. They do not become fertilized. The secondary oocyte won't become a haploid cell or ootid cell unless fertilization and a third polar body. This will not occur from meiosis 2 over here with the secondary oocyte unless fertilization takes place. So if fertilization doesn't happen, this is what's going to get shed during menstruation, along with the uterine lining. So anyway, when fertilization takes place, then we're going to get another polar body, and then the haploid ootid, which is now a zygote. So we'll look at that as well. So meiosis, again, two cell divisions that occur one right after another. In meiosis one, we do have crossing over occurring, as I mentioned. We'll look at it in a little more depth, but in essence, we can take a small segment from one of these chromatids and put it onto another segment of a different one. So we can change up some genetic information. Again, this will ensure diversity among those haploid spermatids. So again, after my when we get to from mature oocyte or spermatocyte, and then that division from that occurs in meiosis one, we half everything. So we have haploid cells here. So that's what this is talking about, the daughter cells. And then meiotic division two, again, is going to reduce the number of cells from two, not reduce, reproduce each of the two cells and form four cells. Hey, these are the haploids. And again, this is going to take place to promote diversity amongst each of these cells. And again, the meiotic two on the oocyte side is only going to occur if fertilization is taking place. So we'll look at that in a little more detail here in a second. Spermatogenesis, primary process where a sperm cell becomes a mature sperm cell. Again, one primary spermatocyte forms two secondary spermatocytes. Each of those will have 23 chromosomes, and then we can divide them one more time, and each of those two secondary spermatocytes form four spermatids, and these four spermatids will each have 23 chromosomes as well. Oogenesis, again, very similar situation going on here. We've got mitosis. Oh, and by the way, spermatogenesis begins at puberty for the male. Oogenesis generally is set before birth or right around birth. We go from, I don't know, maybe 5 million eggs, 7 million eggs in the womb while you're in the womb. And then by the time a female is born, they're down to maybe 1 to 2 million eggs. So anyhow, those are you won't produce any more eggs. In At least they're pretty sure. Now, there's been research continually occurring regarding uh, uh, reproduction and fertility, and there are a couple studies out there that are showing that it's possible that oogenesis or even the development of an egg or more eggs is not ended at birth. So there's a possibility it is happening throughout life. But anyway, there are studies that are showing that as a possibility in mice. So anyway, um, so once puberty occurs, we start to see mitosis and of these cells, and then the beginnings of meiosis one. So we're going to see those secondary. Go back. We're going to see the secondary oocyte and the first polar body again at puberty. This occurs, and then we go through meiosis two, and if fertilization has occurred. So meiosis only resumes if the head of a sperm cell enters. Okay. Ovulation, of course, is when an egg, a mature ovum or egg is released from the ovarian follicles. Insemination will occur in the fallopian tube or the uterine tube, usually in the outer third of the oviduct. So basically where the egg and fallopian tube are connected, that first little kind of curve or dipsy-do is called the oviduct, and it's usually at the outer third of the oviduct as we're becoming a uterine tube or fallopian tube. That's typically where fertilization is going to take place. So we'll look at that as well. Thermotaxis is just a warmer body temperature in that area. So sperm cells are attracted to the warmth. Some fertility specialists recommend taking body temperature for the female if they're having issues with pregnancy, or maybe they're not having issues, they just want to know when they're ovulating. They will take their body temperature every morning when they wake up and see and chart it and see if there's any type of pattern every month where we see a little spike for maybe 2 to 5 days, and then it goes back down. If we do see a spike in internal body temperature around the same time every month, that could be an indicator of ovulation and being fertile. Chemotaxis are chemicals, of course, released that are going to help attract the sperm cells and even trap them and retain them. Sperm cells can live 3 days, maybe even a little longer inside the female reproductive tract. So we've talked about crypts or little kind of caves, if you will, or depressions, pits within the epithelial lining throughout the reproductive tract of the female. And sometimes sperm cells will get kind of held on to until ovulation occurs. So just because the female is not ovulating doesn't mean that they aren't going to get pregnant if they've had intercourse before ovulation. Those sperm cells can live for 3, maybe even up to 5 days. And then when ovulation occurs, then the sperm cells can get released from the crypts or the pits and head into the fallopian tube. So anyway, there's probably a window every month of between 4 and 8 days that a female is most fertile. Okay, So anyway, that's that. Zona pellucida is that outer covering of the ovum that the sperm cells are trying to burrow into. And once one sperm cell makes it into that cytoplasm, we're going to see a chemical reaction that shuts that zona pellucida off and traps any sperm cells that might have made it that far and destroys them. Any sperm cells out here are going to be destroyed likely. So once we have penetration through the zona pellucida, that's it. So, and again, I encourage you to go into Padlet and watch. There are a couple really nice fertilization videos in there. So anyway, spermatozoon reaches the ovum surface, their membranes fuse, and we get the 23 chromosomes in the head of the sperm, 23 chromosomes in the egg, combining and blending. And at this point, we've now got a zygote that is genetically complete. So again, this prenatal period occurs at conception and continues until birth. We're going to see initially what's called cleavage of the zygote and then ultimately implantation. Once the zygote is formed, it's going to continually divide. And that first 3 days or so, we're going to have what's called a morula. And that morula is just a bundle of cells that are rapidly dividing. We're going to see the beginnings of what we call a blastocyst occurring. The morula is a solid a solid sphere, a solid ball. It's the egg and sperm together going through their first 3 days or so of blending genetic information and going through cell division. Again, this is all taking place in the fallopian tube. At the blastocyst, once the morula is, after about 3 days, we start to see a hollowing out of the morula, and we end up with what we call a blastocyst. Remember, blast in cell talk has to do with birth or building something. So we're kind of blasting out the center of that morula and building, starting the process of building what will ultimately be the amniotic sac and part of the placenta uterine lining. Okay, so we'll look at that as we go. So the blastocyst is going to start tumbling down and leaving the fallopian tube, heading off to the uterus for implantation. Okay, so here's a nice little illustration of that. We've got ovulation and a discharged ovum. And again, it's going to kind of linger in this area maybe for a couple of days is all. So if a sperm cell was trapped in a crypt before ovulation and then held there for 3 days or so, ovulation occurs, the sperm cell gets released. A day or two later, we could have fertilization occurring. So if fertilization doesn't occur, the egg will kind of linger maybe for another day or two. So you may have a total of a 4-day period at the most that egg is going to linger in this area. Okay. So let's see, fertilization, and then we've got our first mitotic or cell division occurring, dividing zygote and leading us to what we call the morula stage, where we've got this solid ball of cells. Again, 3 days, it's going to be in that phase or stage, and then as we start to head out of the fallopian tube into the uterus, we now have a blastocyst. And that blastocyst is, again, going to become the yolk sac, the amniotic sac. It's going to enable the binding of the fertilized egg with the uterus, forming a placenta. So anyhow, these are the first, this is the egg getting fertilized, and then the first mitotic division. So we have our morula here. Okay, and then that morula again starts to hollow out, and we get this inner cell mass, which again will become yolk sac, amniotic sac. The trophoblast is the outer wall of this little blastocyst, and that's what's going to embed itself or connect itself into the lining of the uterus. Okay, so the trophoblast, the outer wall, inner cell mass, again, the yolk sac and amniotic cavity. That amniotic sac, of course, is going to help with shock absorption while the baby's in there. It's a nice little watery environment that it lives in. And then, of course, the placenta and the amniotic or the umbilical cord for the baby, all providing food and nutrients and a nutritive bridge, it's called. We also see some of the venous return out of the placenta back into the mother's circulatory system. We see some of the fetal's fetal waste picked up by that umbilicus and the placenta. So anyway, the placenta secretes a large amount of what we call HCG, and that HCG is what we're going to see providing a pregnancy positive or negative if it's present in certain amounts. Yeah, so we can see the mom's uterine lining, and then we can see the placenta embedded, and then the umbilical cord heading into baby. So the purple represents HCG levels. So we see that first trimester, so roughly the first 12, maybe 13 weeks or so. Yeah, because it's roughly 39 weeks divided by 3. So it gives you about 13 weeks for each trimester. We're going to see that that human chorionic growth hormone is at its peak. And again, this is usually when periods are missed and pregnancy tests are taken. So high levels of HCG will indicate a positive test. And that's coming from the placenta. We also have progesterone and estrogen levels increasing after fertilization has occurred. And we see that coming from the ovaries. And then we'll start to see some progesterone and estrogen coming from the placenta as well during that second trimester. And then by the third trimester, the ovaries have stopped producing and releasing estrogen and progesterone, and we see that placenta pumping it out. And this is going to be a trigger as well for pitocin or oxytocin, I should say, from the posterior pituitary to start getting released once we get to 39, 40 weeks. These levels are going to plummet at birth. Okay. A couple other hormones, cortisol levels are going to be at an all-time high during that third trimester. And again, that helps with a lot of inflammatory properties. We see aldosterone helps with sodium and water retention. Moms will definitely tell you that, or pregnant mothers will tell you that they start to retain a lot of water. They gain weight. That especially starts to get prevalent during the second trimesters and then on into the third. And prolactin, another one that we've talked about before, helping with milk production, growth hormone levels. Notice pretty much stay consistent throughout. Thyroid levels go up a little bit as well. Free thyroid hormone going to stay the same. Okay. So anyhow, kind of a cool graphic, a couple of graphics to show you where things are going. Again, at 39 weeks, we're going to get a baby, hopefully. Hopefully it doesn't go too much past 40 weeks or so. The embryonic phase again is really going to start at I know it's a zygote at the beginning, and that process from fertilization to implantation takes about a week to 10 days. Remember, it's 3 days in that morula stage. So anyhow, the first 8 weeks are called the embryonic stage, and then weeks 9 through 39 we call the fetal phase, or we said week 8 to 39, but after week 8. We can see the little being, the first 12, 13 weeks. We see some pretty rapid growth during that second trimester. And then, of course, the third trimester, they start to pudge out a little bit. Okay, and then you've all heard the term stem cells. We have different types of stem cells. Again, they're unspecialized that becomes produce specialized cells. We have what are called potent levels of potency, totipotent, pluripotent, and multipotent. These are the most basic primitive types of stem cells. They're going to be found in the zygote. So this is going to be that first maybe week or so. We have a high level of totipotent stem cells, and then we continue and kind of transform into that pluripotent stem cell grouping, much more specific. So this is maybe, again, from fertilization until maybe 3 to 5 days in. And then at that embryonic stage, once we're kind of getting out of the morula stage and into the blastula stage, we start to see germ layers forming. So we'll look at what there are 3 germ layers. Germ doesn't mean something to do with bacteria or a pathogen. Germ has to do with germination, which has to do with fertilization, growth, development, that type of thing. So we're going to see 3 primary layers of the embryo that are going to differentiate, divide, and become organs and organ systems. And then the pluripotent stem cells, what we call embryonic stem cells. So again, that's that from basically right around just after fertilization until maybe about week 8. So that's where we find the embryonic stem cells. And then the multipotent stem cells are what we all have. Those we see especially with epithelial cells on and throughout the body, because we know those types of cells particularly go through a lot of mitosis and cell division. So what are these germ layers? We have 3 germ layers, what we call the endoderm. And again, this is the embryonic phase of development. The endoderm is the inner layer of cells that are going to ultimately become respiratory and GI. We see the ectoderm is the outer layer. Endo, of course, means in. Ecto is to do with outer. So the nervous system, the epidermis, those outer aspects of the eyes, of the corneas, lenses, okay? The teeth enamel. So outer layers. Okay, and then the mesoderm is the middle layer that's in between all of this stuff. That'd be the dermis, a lot of your connective tissue. Okay, so this is going to be a lot of epithelial tissue, connective tissue. So anyway, bones, muscles, cardiovascular, urinary, endocrine, reproductive, is all in the mesoderm. So this gives a nice little representation. We can see the germ layers here. If you study embryology at any point, you really get into the details of what each moment of embryonic development, day by day by day, basically the first 8 weeks, 30 days, first 200 days or so of embryonic development. we can look at a section of each phase and see and even in each area of that embryo and get an image of what is forming. So these are where we see those distinct germ layers, that ectoderm or outer derm, brain, spinal cord, some of the nerves, and certainly the skin. And then the mesoderm portion or grouping of cells, we can see quite a bit of action going on there. That's going to form basically all of the skeletal muscle, the cardiac muscle, the bones. Okay, you see the kidneys, and then the endoderm, primarily relating to respiratory and digestive. So anyway, I don't want to spend too much time on most of this. We're just about done with this chunk, and then we'll talk a little bit about genetics and heredity, and that'll be it. So cortisol, remember cortisol levels are definitely going up, and as they get kind of to a peak out, we're going to see human chorionic growth, human chorionic gonadotropin hormone slow down, and then that's going to remove. We also see progesterone levels start to drop. This is going to take the brake off of oxytocin up in the posterior pituitary, and then that's going to stimulate uterine contractions. And again, this oxytocin is clinically known as pitocin. So, and then we're going to have our stages of labor. Stage one, we're going to get those first contractions that occur where, as oxytocin is released. We're trying to get that cervix dilated. Stage two, we're going to see that maximum dilation. So it would be at a 2, and then a 4, and then a 3, or 5, and get up to like an 8 or a 10, and then we can start seeing some action taking place. And then stage three, we're going to get the placenta to be ejected or expelled from the vagina. Okay, so stage one, contract. Stage two, get that baby through the cervix and through the vaginal canal. And then stage three, we need to deliver the placenta. Multiple births, we have maternal or identical twins. That's when we've got a single zygote that's split early in development. And then fraternal twins, we're going to have two different eggs. So this is one egg that split, one fertilized egg that divided. And then this one is we have two different eggs that were fertilized by two different sperms. So two completely different buggers growing in there. Okay, fraternal versus identical. Okay. Again, we see identical. We've got an egg, it gets fertilized, and then at some point we see that yolk sac and that amnion, what's going to be the amnion, they divide. And so then we'll get that trophoblast or that blastocyst to divide. Okay. And they are probably going to be contained within that same sac. But they do have a little bit. So they have two separate amniotic cavities, one placenta. Whereas these buggers, we're going to have two eggs, two sperm, two sacs, two placentas. So if you've got triplets or quadruplets or quintuplets, you're going to have 5 placentas in there. IVF and other fertilization specializations, fertility treatment lead to multiple births that are going to be of the fraternal variety. All right, so let's look at genetics briefly here. I don't want to spend a heck of a lot of time on this, but why does baby look like they do? Well, they got a genetic code from mom and dad. They're little what are called chromatids, chromosomes. So we'll look at that a little bit. We've got chromosomes are the compact form of DNA, and that's what's going to end up getting split. But we can see all these nucleotides forming and combining together, and then we get those condensing, and we get these little histones, these little wheels forming what are called nucleosomes. And then we can start winding these little spools or these histones and nucleosomes together. And then we get some chromatin. And then we start winding the chromatin, and at that point, we've got chromosomes forming. We can see these chromosomes winding and winding and winding, and then we have a complete mature chromosome here. Okay, so each gene is a sequence of those nucleotides, A, T, C, and G, that we've looked at in the past. And again, the genetic code will determine structures, it'll determine functions. We did the Human Genome Project in about 92 and concluded it in '03. So about a 10 to 12 year project, 19,000 genes and non-coding DNA. Some of that non-coding DNA might be what we call junk DNA or DNA that doesn't really do much of anything. So anyhow, we see genes encoding proteins. We look at transfer RNA, messenger RNA. And so we've got all of these 23 pairs. And again, that 23rd pair is the sex determinant or gender pair. And you can see this is kind of cool. We look at a chromosome up close, and these are what they call sister chromatids. So it looks like it's a complete X, but these are actually held together by what we call a centromere. And the centromere is kind of like a belt that keeps the two sister chromatids combined. And so we can take a little tiny chunk and analyze all that chromatin, and then we can take that chunk even further, and we can get the gene sequence of just one little strip. So what they've done here is they've taken this little tiny strip and they've made what we call an ideogram or ideogram. And then they take that little chunk, and then we can see all these A's, T's, C's, and G's. So this is the gene sequence for this particular spot on this particular chromosome. It could be any one of these. So anyway, that's called genomics or genomics when they analyze DNA. Proteomics looks at the entire group, the entire genome. Again, they've got these ideograms or ideograms, kind of cartoony look. Um, and again, we're looking at these nucleotide bases, A's, C's, T's, and G's. And we've, again, meiosis, we've seen that, 23 from mom, 23 from dad, giving us 46. Independent assortment, that's when we're going to get some genetic variation is basically what they're saying. And how can we get genetic variation? Well, by doing meiosis or meiosis, we can get it crossing over as well. So let's look at independent assortment, and you can see just how many, there's a zillion different possibilities. So we've got, again, the diploid parent cell, and then we get that chromosomes aligning, and then we're going to divide those 46ers into two 23s in meiosis 1. Now, this is showing just one example. We've got 4 items that we can work with. So we can take a big blue and a little blue, put them over here, and a big red and little red, and put them over there. We can also take a big blue and a little red, like we see over here, and so on. We have 8 different options minimally, probably. Uh, uh, and from there, we can divide these then up into, you can see the hapless. We're going to slice these in half, and then we're going to get a little chunk over here. We're going to slice another one in half, and so on. And one chunk here and one chunk here may end up over here. This chunk here can end up over here. So we have 4 different possibilities from this little batch here. Change it up. Okay, so now we've got a big blue and a little red. So we're going to have a different group of haploid gametes. These are, again, individual sperm cells. That's one way that it happens, and that's not even the half of it. This is the half of it, what we call crossing over. At this point here, we can even take a little chunk out of here and move it over to this guy. So that's what's going on here. You can see they're uniting and basically trading chunks. So you can imagine you'll never get the same exact combination from a sperm cell or a haploid sperm cell. Let's see. So you're going to inherit those traits, one from each parent. So the dominant gene is what's going to be seen, capable of masking the effects of recessive genes for the same trait. If recessive genes' effects are masked by the effects of a dominant gene for the same trait. So we're going to see dominant traits and recessive traits that get expressed. We call that what we call the phenotype. So we'll look at that for instance, with albinism. We've got the big, a capital letter always represents the dominant gene. Lowercase letters represent recessive genes. So we've got mom is what we would call it's a heterozygous. Dad is also heterozygous. So if both for this particular gene, and this is just one spot on the whole area. So this particular gene codes for melanin production or melanocyte activity, which helps with pigment. So, so on that gene for pigment or melanocytes, mom is a carrier because she has it's a recessive trait. Dad is a carrier. He's got the recessive trait as well. So when we divide that up, we've got an ovum with a big A, we've got an ovum with a little a, we've got a sperm cell with a big A and a sperm cell with a little a. Again, in that particular sequence on that exact gene that codes for melanocyte activity. So if little a meets up with little a, if this happens to be the two gametes that fertilize, then we're going to have two recessives, a recessive from mom and a recessive from dad, for no melanocyte activity. They call that albinism. Okay. This is what they call homozygous recessive. So homozygous, meaning homo is same zygote, same zygous, homozygous, recessive. Okay, and heterozygous, remember, means different. So if you're heterozygous, the dominant trait is what gets expressed. So dad and mom don't express albinism because they're heterozygous. So they do have they're going to express physically the dominant trait. Okay, but if so they're carriers. So that means if they're with another carrier and it happens so they could have 4 kids, well, say 3 kids. One child or offspring could end up with the 2 little a's or homozygous recessive for melanocyte activity. Another one could get the big A from mom, so the dominant trait, and then the little a or the recessive from dad. If that's the case, they're going to have normal pigmentation because the dominant allele will overtake the recessive allele. However, they're still a carrier. Hey, if we get big A, the dominant allele, and big A, the dominant allele from both, we're going to have a child that has normal pigmentation and is not a carrier at all. So, yeah, there you go. There's a lot of options for the tens of thousands of genetic characteristics that we all have. So the genotype is what we see here. It's expressed as capital and lowercase letters as a combination, and dominant allele is capital, recessive is lowercase. So homozygous, you're going to have two capital letters or two lowercase letters. You're going to have the same identical form of the trait. With heterozygous, you're going to have a capital and a lowercase, with the dominant trait as the capital being what is expressed as the phenotype. And the phenotype simply means the physical characteristic that's expressed from the genotype or the gene. Okay, and again, a carrier is someone who's probably heterozygous, so they're a carrier but they're not expressing the trait. So again, phenotypes are going to be expressed with dominant and so with heterozygous and homozygous dominant, you're going to see the phenotype expressed. Okay. If you have homozygous recessive or homozygous recessives to small letters, you'll see the phenotype expressed. So depending on what the physical characteristic is, again, blue eyes, any eye color other than brown, for the most part, brown and hazel kind of go together. But if it's green or blue, I guess hazel too, even though they have a tint of brown, but basically brown and non-brown eyes. So brown eyes are dominant, anything non-brown recessive. So mom and dad could have big A, little a, and big A, little a, so they're both going to have brown eyes. Okay, but as we saw over here, so if this is for eye color, this guy's going to have brown eyes. This gal's going to have brown eyes, but this little bugger could have blue eyes. So two brown-eyed parents could have a child with blue eyes if the two recessive traits are what show up as the phenotype. Yeah. Polygenic traits, a combo of several different genes, co-dominant when two different dominant genes occur together, having an equal effect. Two brown-eyed parents sharing the dominant traits. Okay. Let's see, sex-linked traits. Again, female chromosome is the X, the male chromosome is the Y. So again, we see the mother's sex trait. So the 23rd pair of chromosomes is the the gender of the individual, and we would see that her 23rd pair of chromosomes for gender are going to have double X. Okay, the male is going to have an X and a Y. So notice no capitals or lower cases. So we're just sticking with X's and Y's. So mom is going to release an ovum that's going to have one X going this way and one X going this way. Which of these is going to get fertilized? I don't know. So we do know that that the female doesn't determine the sex of the child, because it's always going to be female. The male, however, with his X and his Y, the X splits. If the X comes together with an X, we get a female offspring. If the Y comes together with an X, we're going to get a male offspring. So the male does determine the sex of the child. They research, can we tell the difference in sperm behavior, sperm motility, anything to do with sperm? Do we see gender differences regarding the sperm cells, their behavior? Are they there's some research showing that male sperm cells tend to be more sprinters, female sperm females, I keep saying male, the female sperm cells more of like a long distance runner. Male sperm cells are more sprinters. So if they're highly energetic, the female is at a peak fertility. That may lead to a boy. Slower pace, longer lasting, possibly those sperm cells that tend to take their time getting to the egg. So maybe we're not at ovulation or peak fertility. If we're at a day or two before, the female sperm cell representation may be the one that lasts and hangs out and waits those couple of days a little bit easier than the male. So anyway, that's all being researched. I don't know if there's anything definitive on being able to tell what's what. And then we can look at sex-linked inheritance too. So we've got mom with an abnormal X chromosome. So it could have to do with color blindness. So the one spot on this particular ovum, and that has to do with cones and the alignment of cones in the retinas, comes from the female side. So moms determine whether or not maybe son has color blindness. Males typically are more prone to being colorblind. Partially because on the male, on the female, from the male side, this doesn't have the colorblind mutation. So this may be what dominates. So she's going to be a carrier, but she's not going to express color blindness. Whereas this bugger doesn't have the non-colorblind X chromosome that's going to help to offset the mutation. So he's going to be colorblind. Yeah. So anyway, there's genetic mutations. There could be a deletion of a base sequence, a nucleotide. We could have a nucleotide that's in its usual spot and then gets deleted out and replaced. Mutagens are agents that cause mutations. A lot of these are environmentally associated. We are looking at something called epigenetics as well. Non-disjunction, where we may have, instead of splitting down here, we end up with an offspring that is trisomy issues. So we can look at mitochondrial DNA. Mitochondria have their own DNA, and we can find the map of mitochondrial DNA, and we know where mutations are located that lead to certain diseases or disorders. So anyhow, the rest of these are just different genetic disorders. So we know cystic fibrosis, chromosome pair 7, recessive genes having to do with mucus in the lungs, mucous membranes in the lungs, PKU, again, another recessive gene trait that doesn't allow what's called a hydroxylase for phenylalanine. So phenylalanine deals with, is a type of amino acid or protein that has tyrosine in it. So anyway, phenylalanine can't be converted into tyrosine without this particular enzyme. So if you don't have this enzyme, phenylalanine will accumulate in the body and cause PKU, phenylketonuria. Okay. And that can lead to brain tissue destruction, too much phenylalanine. So one of the main treatments is diets low in phenylalanine. This is congenital. You would be born with it. Okay, so here's a couple of, so basically PKU means you have to avoid all of these: dairy, meat, cheeses, eggs, nuts, legumes, aspartame. You should avoid that anyway, PKU or not. This should be banned, frankly. Beans even. Ooh, well, no. Looks like they show those are peas, however it looks like to me. Sweet potatoes, unfortunately, fall under high phenylalanine group. Sweet potatoes are extremely healthy unless you have PKU. And then we've got most vegetables, most fruits, pastas are going to be good for that. So anyway, here's what you need to avoid if you have PKU issues. Okay, that's it. The rest of these you can certainly go through and review on your own. Epigenetics deals with environmental issues or environmental toxins that cause a variety of different mutations. Chromosomal diseases, we know Down syndrome or trisomy 21. So we've got the 21st pair. There's a triplet instead of a pair of that chromosome 21. Cancers, possible link to genetics, abnormal genes. They think roughly 15%, maybe even a little less, of cancers can have a genetic basis. That means most cancers have an where'd it go? I don't know where it went. Epigenetic basis. There it is. Ooh. So epigenetics we're seeing probably more likely than having coming from your ancestral DNA. So genetic counseling and can help with understanding, maybe preventing Punnett squares and pedigrees. So pedigree, we think about this in like relation to dogs and whether it's a pure pedigree or not. Punnett squares, again, we can analyze specific genotypes. And so again, this one's for PKU. This could be for eye color. This could be for any number of phenotypes. So a Punnett square, we create a 2x2. We've got big P, little p on the top. Dad has big P, little p, so we can determine what are the possibilities for the offspring to have PKU, to be a PKU carrier, or to be PKU free. So we've got a 1 in 4 chance of big P, so that would be clear normal. We've got a 50% chance. So we have big P, little p, big P, little p. That's going to be a 50% chance of a PKU carrier. So they're not going to express it. And then we've got a 25% chance of little p, little p. Yeah, so 1 in 4 chance if mom and dad are both carriers of the offspring having PKU. So this just shows other, again, these could be for anything, hair color, eye color. Okay. Gene therapy, gene replacement therapy. Okay. All right, so that's it. Amniocentesis, we can get an idea from the amnion or the amniotic fluid. We might be able to ascertain some of what's going on with baby and if they're going to possess some heredity or congenital disorders. Okay. Just threw this in here. We've talked about amino acids quite a bit. It's a nice little chart. Here's phenylalanine. Gets converted to tyrosine in the presence of that hydroxylase enzyme. Again, if you don't have the enzyme, phenylalanine will build up and can cause some brain issues. We know with amino acids that we have non-essentials, so these are recycled in the body. As you can see, tyrosine is non-essential because we can, if we're eating enough phenylalanine, and we have the enzyme, we can convert that into tyrosine. So that's it. If you have questions, please let me know, and have a great day.